Part B of Chapter 13, we're going to continue looking at the life cycles of the viruses. We left off in Part A looking at the DNA virus of animal virus replication. So we see that the virus is going to attach. Again, entry into the cell first has to be uncoated, and then we see the release of the viral DNA and the capsid proteins. The viral DNA has to get into the nucleus in order to make messenger RNA. This is then going to direct the making of the viral proteins. The biosynthesis of viral DNA is being replicated, and then in late translation, those capsid proteins are synthesized, and we start to put the virin particles back together, and they are released from the cell. When we talk about the biosynthesis of the DNA viruses, we can have both single-stranded DNA viruses and double-stranded DNA viruses. Both of these have cellular enzymes that transcribe the viral DNA in the nucleus, and so they're pretty straightforward in how they replicate. For the RNA viruses, however, we have what are referred to as single-stranded RNA positive strands, single-stranded RNA negative strands, and double-stranded RNA viruses. When you have the single-stranded RNA positive strands, it is the RNA acts as the messenger RNA. So it is immediately going to drive the making of RNA polymerase, which then is going to synthesize the opposite strand so that it can make more viral messenger RNA in the cytoplasm. For both the single-stranded RNA negative strand and the double-stranded RNA, they have to already bring in an RNA polymerase. So they either contain an RNA polymerase or bring it in as a capsid RNA polymerase in the case of the double-stranded RNAs, but they are then going to need that polymerase to then make the viral enzyme copies of the viral RNA to then make the messenger RNA. So this means that it's just kind of the same thing happening, but at different entry points, in whether it's actually the messenger RNA or it has to make it first. The take-home message, though, is that DNA viruses are going to replicate in the nucleus, RNA viruses are going to replicate in the cytoplasm. So whether it's a positive strand of a single-stranded RNA virus, a negative single-stranded RNA virus, or a double-stranded, all of them are going to replicate in the virus. It's just the positive strand is actually going to be able to make, or it acts as the messenger RNA, so it's going to be able to immediately make its RNA polymerase. The other ones have to bring in the RNA polymerase and then copy over to make message. So the other thing that kind of goes along with this is that some viruses have a unique enzyme, reverse transcriptase. You've probably heard about reverse transcriptase because of HIV, but just to remind you what this enzyme is able to do, when we talk about the central dogma, we basically say that DNA is going to be transcribed by RNA polymerase into messenger RNA or other species of RNA. So it's DNA to RNA. That is supposed to then flow directly into messenger RNA being made into protein, or the RNA goes and does something functional if it's a transfer RNA or something. So we do not have in our cells reverse transcriptase. We can only take things from DNA to RNA. Some viruses have this unique reverse transcriptase enzyme that allows the RNA to be back copied into DNA. So again, the classic example is the Retroviridae, for which HIV is a member, where it is going to have viral enzymes that copies the viral RNA to make DNA in the cytoplasm. That DNA then moves into the nucleus. But we have other things like the HepDNA viridae. Hepatitis B is an example that you're familiar with. So this one, it's using cellular enzymes to transcribe the viral DNA in the nucleus, and then uses its reverse transcriptase to make copies of the messenger RNA, using that to then make viral DNA, and the process is then going to continue in the life cycle. So if we look more specifically at a retroviridae life cycle, similar to HIV, we see that two identical positive strands of the RNA are being brought in. So the first thing that's going to happen, it's obviously going to be endocytosed in. The capsid protein coat needs to be removed. So you have the release of the viral RNA and the reverse transcriptase and things like integrase. So the first thing that happens is that the viral RNA, using its unique viral reverse transcriptase enzyme, is going to make viral DNA. That viral DNA is now going to go into the nucleus and integrate into the host genome as a provirus. That provirus is then going to direct the cell to start making viral proteins. So it'll direct that messenger RNA is made, viral proteins are going to be made, and then it's going to go through its biosynthesis and maturation to make complete virus particles, and those are then going to get out of the cell. So we see an example here of one that is budding out to complete the life cycle. The problem with proviruses, though, is that in animal viruses, proviruses never ever come out of the host genome. So they are not like a prophage in that way. Prophages were able to go in and out. That's why if you have these integrated viruses, we say there are some viral infections that you can never be cured of because that provirus has integrated into the host genome, and therefore it's going to remain there. It may not always be expressing, so it can remain latent and not be making new virus, but more than likely at some point it will be expressed and start to make or shed new virus. The other problem, you just don't want things integrating into your host genome. Oftentimes these are associated with tumor events, so it converts the host into a malignant cell, often because it hits an area that is going to control growth, and therefore you end up having a malignant event. So we see that virus and cancer are related. So when we talk about the consequences of viral infection, not only do you have that it can cause like a true infection and cause a lytic infection to get back out of the cell, we have other things that can occur, things like transformation to make a tumor cell that we'll talk about now. Oncoviruses contain genetic material that integrates into the host cell genome and can cause cancer. Cancer is multifactorial, so it's only about 10 to 15% of all cancers that are known to be virus-induced. However, that is still significant. The first of this was demonstrated back in 1908 in a chicken leukemia. Then in the 1930s, this was established to also be true in a rodent model, but it wasn't until 1972 that this was demonstrated to actually occur in human cancers as well. It was a little hard to understand why viral etiologies, viral causative agents could cause cancer. It was unrecognized because, first of all, most viruses do not cause cancer, so this was a relatively rare event. The cause and effect, cancer may not develop until long after that infection. So to attribute that cancer to a viral infection that may have happened possibly decades before was quite difficult. And I think most problematic is the fact that cancers are not contagious. When we think of viral diseases, we usually think of them being passed around, and so therefore the fact that cancers are not contagious seemed a little odd for something that could have a viral etiology. But we do recognize that viruses are associated with cancer. Activated oncogenes can transform normal cells into these malignant cells. There's actually the proto-oncogenes, normal, non-mutated forms that probably have been derived from past ancestral genomes. So these types of cancer-inducing genes, they are out there and they get put into viruses and are moved around, and when they then integrate, they transform the cell. Typically what happens in a malignancy is that you have uncontrolled cell growth. So these cells are not only growing uncontrollably, they also kind of forget what they're supposed to be. They become less differentiated. They also lose their contact inhibition, so they no longer recognize as in a organ or a tissue. They only grow to a certain size because they know their neighbor is next to them, and they're all supposed to fit into the liver, and so this is what a liver cell is supposed to be looking like if it's differentiated. As cells become transformed, they lose that contact inhibition. They just grow over top of each other. And oftentimes this causes the expression of certain antigens that are associated with that malignancy. So the genetic material of these oncoviruses become integrated into the host genome, and they're not going to come back out. A couple of examples of oncogenic viruses that you may have heard of. The herpes viridae, so the Epstein-Barr virus, and we see that that can be the causative agent of what's referred to as Burkitt's lymphoma. The one most people probably know, the papillomavirus, because of its association with cervical and uterine cancer. HPV-16 is responsible for about 50% of the cancer-causing strains of the papillomaviruses. The hepidna viridae, the hepatitis B, the acute infection is not all that problematic. The problem is usually when the virus becomes chronic and then you start to have liver cancer. Some of the adenoviruses and pox viruses can also cause cancer. Oncogenic RNA viruses are in the Retroviridae family. So we see things like the T-cell leukemia and lymphomas, sarcoma viruses that are common in chickens and cats and rodents, and you've probably heard of feline leukemia as well. So we see both RNA and DNA oncoviruses. So those proviruses are pretty important for the fact that they integrate into the host genome and never ever come out. So they can cause disease, they can cause cancer. So we'll look at some of the other transforming properties as well. So talking about those other outcomes, latent and persistent viral infections are kind of when you have those integrations and what happens in these types of infections. So we'll talk about that in the following slide. Latent viral infections, again, the virus is able to remain not expressed. So it is basically latent or hiding out, so people remain asymptomatic for long periods of time. Classic example is the herpes viruses. There's a large number of people that are carriers, but only about 10 to 15% of people actually express. You can have herpes type 1, which is the herpes simplex virus that is associated with cold sores or what people refer to as fever blisters. Or you can have things like the chickenpox virus, the varicella virus, that is, when it comes out of latency, causes shingles. Immunosuppressed individuals cannot handle these latent viral infections very well, and unfortunately, they are often fatal to people that are immunosuppressed or compromised in some way. Persistent viral infections, on the other hand, are kind of a slow, gradual process. When we see these persistent viral infections, they almost always are neurological and almost always fatal. There are low levels of the virus that remain undetectable, and it gradually builds up over time. But when it does, it all of a sudden spikes, and usually that is when you have some neurological event, and again, usually that leads to fatality. One example is subacute sclerosing panencephalitis from measles virus. Young males that get measles have a 1 in a million chance of getting this persistent viral infection, but if they do, it's almost a certain death sentence in about 10 years after they had the measles. So if we look at the illustration graphically, we see that for acute infections, you see a very quick peak that then goes down. Hopefully we have cleared the virus. For a persistent infection, it means that we didn't completely clear it, and so you maintain these low levels for a period of time, but at some point it starts to increase, and that's when you have usually a neurological symptom that is going to occur, and again, these are almost 100% fatal. With latent infections, appears like it's gone away, it's hiding out in the genome, but then you have these peaks. It doesn't have to be just one peak. You can have that this goes into periods of remission for a while, then starts to express. And so the latent viruses can have a cyclic nature where they have periods of latency and expression. So some examples of persistent viral infections, as we just said, the subacute sclerosing panencephalitis from the measles, but there's also a progressive encephalitis from rubella, which is the German measles. HIV, we believe some of the AIDS dementia complex may be part of the HIV being a persistent viral infection, and we see some of the enteroviruses as well can be associated with mental deterioration that can be associated even with AIDS. So when you talk about a classic latent viral infection, the thing that probably comes to mind is the relationship of chickenpox to shingles. You first get varicella, the chickenpox, as the primary infection, and you bloom out in these kind of vesicular blister-like lesions, but then you have the virus. It goes into the dorsal root ganglion of the spinal cord and just hangs out and goes into a latent period. At some point later in life, either you have some event that activates it, failure of the immune system, more common to happen in the elderly. You have a reemergence of the virus, now that is referred to as the herpes zoster, and because the nerve split off of the spinal cord to the left and the right, you only end up having the lesions unilaterally on one side. The thing that is the most symptomatic about the shingles is that it has what's referred to as these post-herpetic neuralgia. Very, very painful if you know anybody that has had it, but this is kind of a classic latent infection that then reactivates. So antiviral drugs are difficult to try to develop. The virus has basically hijacked the host cell, and so trying to target so that you do not have toxicity to the host cell is problematic. So what can you do? Think of the life cycle, and that kind of lays out the targets. You could first try to prevent the virus from attaching to the host cell. That would obviously have to be a very early event. Then if you didn't prevent the attachment and the virus is now trying to get in, prevent that penetration. So prevent that endocytosis from occurring. All right, it got in the door, don't let it take its coat off. Again, it's a little bit further in the life cycle. If you let it take its coat off, try to inhibit DNA or RNA synthesis or reproduction. However, that obviously is going to have consequences for the host cells, so that is a little bit more problematic. Somehow prevent the assembly of the new viruses, and last-ditch effort, don't let it get out of the cell. So all these are potential targets, but developing these as true antivirals is not simple. So hence we see with things like COVID-19, we just don't have a plethora of options there at the ready. It takes a while to develop these drugs to the point where they are effective and also safe. One of the go-tos, and will be our classic go-to, is acyclovir. This is what's referred to as a nucleoside analog. So when you're making DNA, you have the phosphate, and that is going to be coupled with a nucleoside so that you get a complete nucleotide. That, in the example here, we have a guanine nucleotide that DNA polymerase is then going to insert anytime it needs the G in the growing chain of DNA. Acyclovir is what's referred to as a false nucleoside. So when the phosphate combines with the acyclovir, you end up having the inability of the DNA polymerase to continue making the DNA chain. What acyclovir looks like, it looks very much like the guanine, but you see the ribose ring for the guanine that is going to allow the backbone to be made. With acyclovir, it doesn't have the ribose ring, so it can't connect the nucleotides one to the next in the growing chain of DNA. In this case, it is then going to block the DNA replication. We now see that there's many different derivatives of acyclovir, but all of them have pretty much the same mechanism. There are other antiviral agents, things like protease inhibitors and reverse transcriptase inhibitors. Many of these have been developed because of the AIDS epidemic. So the protease inhibitors prevent viral replication by selectively binding viral proteases that are needed to be able to allow the reproduction to occur. The reverse transcriptase is unique to the virus. So that would be a non-toxic target. So that's why people were very excited about making the reverse transcriptase inhibitors. However, we have learned that it's not going to be a one-drug therapy that works on something as complex as HIV. We've had to develop what has been referred to as highly active antiretroviral therapy, or HAART. There's other acronyms for this, but basically it means you have to utilize a combination of these antiviral agents to be able to try to keep ahead of the virus. The other problem, the virus mutations, means that often the virus becomes resistant. So people that are maintaining their HIV status without progressing to AIDS oftentimes have to have a combination of many of these antivirals and keep changing them to stay ahead of the virus. There are some new therapies. These fusion inhibitors would be preventing that first contact. And so preventing the entry of HIV into the cell would be a great way to prevent infection, but it needs to happen early. You would have to know that someone needs to be on these types of therapy. It's relatively expensive still, and it is only available as an injectable product that needs to be injected twice daily. There's also some integrase inhibitors. This is going to prevent the provirus from ever forming by integrating into the host genome. So if you can prevent the formation of that provirus, then obviously the virus is not going to be able to stay latent within the host genome. As I said, much of what we have developed has come from the AIDS epidemic. So this is a list of just some of the HIV drugs. So we see the nucleoside, nucleotide, reverse transcriptase inhibitors, some non-nucleoside reverse inhibitors as well, protease inhibitors, integrase inhibitors, and some of the newer pharmacokinetic enhancers, as well as those fusion inhibitors and inhibitors of entry. So these are going to be the classic antiretroviral medications. Other antiviral drugs, we talked about things like Relenza and Tamiflu that inhibits the release of things for the flu because it's acting at the neuraminidase spikes. The adamantanes inhibiting uncoating, but we've learned that it's not a one-size-fits-all. One therapy that is being used, interferons are proteins that talk in the immune system. They basically, you hear about things that are like inflammatory cytokines that are going to get the immune system kicked off. These interferons, there are a category of them that prevent the spread of virus to a neighboring cell. So these have been used in things like controlling viral hepatitis. So the first cell is going to be infected, but this prevents the virus from being able to spread to neighboring cells. Because it's sometimes hard to control these interferons, they also, since they're proteins, they don't stay around for very long in circulation. So another approach is to use imiquimod, which promotes the interferon production naturally. This has been effective in controlling genital warts. Although it's not really a virus, prions are talked about in this chapter as well. So we see with this prion hypothesis that a normal protein gets converted into a bad disease protein. So what does that actually look like? When we talk about prions, they got their name from proteinaceous infectious particles. People really doubted that these protein particles could actually cause disease. It was first proposed by Stanley Prusiner back in the early 80s, and he did receive the Nobel Prize for his efforts. They cause what's referred to as transmissible spongiform encephalopathies. If you look at the histology upon death and are doing the pathology of the brain tissue, it literally looks like Swiss cheese. So it looks kind of spongy, and that's how we got the name of the transmissible spongiform encephalopathies. There are some that have a genetic component. So things like Creutzfeldt-Jakob disease, the CJD, that is going to have both a variant form and one that does have a genetic component. There's also weird ones that run in families. There's an Italian family that is inflicted by fatal familial insomnia, but the one that's probably best known is mad cow disease. This probably came from what is referred to as sheep's scrappies. It apparently makes the sheep itch, so they kind of scrape themselves up against fence posts and things, but it's usually not necessarily fatal to them. The prion hypothesis says that there is a normal cellular prion protein, and it's found on the surface of the cell. We're not exactly sure of its function, but it's normally there. Then we have what's referred to as the abnormal, the scrappy protein form, and this is going to be what results in the accumulation in the brain cells forming the plaques. So what happens is that the disease form, it starts to interact with the normal form, and it gets taken into the cell by an entosome that then occupies a lysosomal compartment. Once it gets in the cell, then it starts to have its toxic effects and kills the cell. So if we look at this life cycle, again, it is a normal protein, but when the disease form shows up, it interacts and basically causes everything to be taken into the cell. And as that disease form of the prion accumulates in the endosomes, some of the contents gets transferred to the lysosomes, and that results in the death of the cell. And that's why these are always going to be fatal diseases. The normal prion protein is relatively easy to control. So it's easily digested by proteases. The form is these coiled alpha helices. They're destroyed by heat, detergents, most of the things that we use to disinfect. The abnormal form, however, is predominantly beta-pleated sheets, which are kind of like an accordion sheet, and they end up getting found in the cell rather than on the cell. They are virtually insoluble, cannot be degraded by proteases, and they survive high heat, radiation, detergents, all of our disinfectants. They're almost indestructible. So that's why people got really worried about mad cow disease, if that really got in our food chain, what that could do, and that we did not have the ability to get rid of it. So how did we end up with mad cow disease? Why did it suddenly appear in British cattle in the 1980s and then go on to become an epidemic? One of the things that we propose is that this actually occurred because we were forcing the cannibalistic practice of cows getting protein supplements from carcasses of other animals. So things at the slaughterhouse that were not ready for human consumption were what's referred to as meat and bone meal. At one time, this had a cooking process and a solvent extraction. In the 70s, they stopped doing those procedures, and people have theorized that maybe this allowed things to become more infective. So we believe this may have contributed to the outbreak in the British population of cattle, and it was declared a new disease in 1986. The second hypothesis is maybe this just arose in one British cow, and that poor cow got processed into meat and bone meal and spread it to other cows. However, we don't really think that's probably the case of what happened. A third hypothesis is that it's just been endemic in cattle for many years, but it wasn't until the change in those processing of their meat and bone meal that it ended up spreading epidemically. Most people pretty much support the first hypothesis, and it is typical that as things jump species, as this maybe went from the sheep form to the cow form, that it did become a more virulent form. Even though a prion is not a virus, it may be acquiring similar capabilities as it jumps species. So viruses are hard to control. It's not that we don't have antivirals, we do. And it's not that the vaccines that we have are just against viruses. There's vaccines against both bacterial agents and viral agents. But we'll see there's a lot of viral vaccines. So this is a good place to talk about vaccines in general. So there's many different categories of how you make a vaccine. There are attenuated whole agents. These are referred to as weakened but live. The advantage is that they are like the real thing, the natural live virus or whatever the agent is, and so they provoke better immunity. The disadvantage is that these could potentially revert back to the more virulent form and cause the disease that they were meant to prevent. The inactivated whole agent is what's referred to as dead. The whole agent is still antigenic, so it can stimulate the immune system. However, it cannot cause the disease. The disadvantage is that it's not as much like the natural version, so it only provokes weaker immunity, and these usually have to be an injectable product, which is problematic because they usually also then require refrigeration. When you're talking about vaccinating the world, these are considerations. We also have toxoids. These are inactivated toxins. So things like Clostridium tetani, the causative agent of tetanus, the problem is the toxin that causes everything to go rigid. If you can cause the immune system to make antibodies to that toxin before it sees it, then you will have protective immunity. So they give people the inactivated toxoid form so that they will have protective antibody levels if they see the toxin. It is best to not have to use the whole agent if possible. So we now have what are subunit and recombinant vaccines. They are acellular, meaning they're not the whole cell. So you're finding an antigenic component, either of a cell wall or a membrane or something that is going to be able to be used to cause the immune system to respond. But when it sees the whole agent naturally, it's going to provoke an immune response that is protective. The conjugated vaccines were hard to develop. So these are a lot of our meningitis and pneumonia type organisms. They have large capsules, and those capsules are made of polysaccharides, which are very repetitive, so they're not very good at stimulating the immune system. Therefore, we had to conjugate them or couple them with a protein. We're basically trying to fool the immune system to react to the protein, which is a good antigenic agent, stimulates the immune system, and by coupling that capsule with it, basically it comes along for the ride so that we are able to effectively make a vaccine. The newest of the vaccines are the nucleic acid vaccines. These use a DNA or RNA as an alternative to those whole agent strategies. They're still being underdeveloped and evaluated, but basically you're taking a DNA type vaccine and you're taking this as a plasmid and trying to put in viral antigens that are going to then stimulate a response. These require things like a gene gun to get them into the cells. No needles or refrigeration needed, so once you have that piece of equipment, pretty easy to vaccinate. These still, they've been around for a couple of decades now, but we haven't had a really good, effective one. And that's why it's a little bit concerning that one of the leading candidates is one of these DNA vaccines for COVID-19. Whether it works or not will be a story still to be told. So one of the things that I hope that you really take away from 226 is to be a proponent for vaccination. As future healthcare professionals, it's important that you educate the public to how important it is for us to vaccinate. Vaccines for children are so important. The CDC estimates that vaccination of children born between 1994 and 2018 will prevent over 400 million illnesses that will help avoid over 900,000 deaths and save nearly $1.9 trillion in societal cost. Those are reasons that are excellent, but it's really about the safety and well-being of our children. Vaccines do save lives. It is very stressing to see the amount of vaccine hesitancy and anti-vaxxers that are occurring these days. This runs the gamut. I don't want to imply that these people are bad in any way. However, we see people that really just don't believe in the process, whether it's people like Poisonous Pete, who just thinks that these are going to poison him in some way, to some people like Naturalist Nancy, who just believes your immune system is good enough, so why should you have to do any type of vaccination to be protected? It's not good enough is the answer. So unless we can, for COVID-19, have enough people that have natural immunity, which is probably unlikely unless everybody gets infected, you have to have people that are getting artificial immunity by being vaccinated. So that's why it's important that we help people to understand. People have not of your generation and really of my generation, they haven't seen things like the measles. So if you see in the graph here that vaccine was licensed for the measles back in the 60s, you had this precipitous drop in the number of cases of the measles almost back down to background levels. So people don't realize what these types of diseases look like. So that's why it's great that we have things like the Bill Gates and Melinda Gates Foundation that are really proponents of vaccination. They have put out there that 7 vaccines in 73 countries have saved more than 6 million lives since 2000. So things like vaccinations to meningitis and our Hib vaccine, things like measles, and a variety of things to rotaviruses and the pneumococcal strains, yellow fever. These are all very important vaccines that are going to be protective of what are potentially deadly diseases in children. It's alarming that we saw last year one of the highest numbers in 25 years for measles. So we had a measles outbreak, and we see here the map of what had occurred in 2019, Pennsylvania being not a hotspot, but certainly still alarming. And you see in the darker colors those areas like New York and California, Florida, Texas, that really had large number of cases of the measles. So what is it? Well, first of all, people just forget that measles just isn't a little rash. That measles is a very serious disease. About 1 in 4 people get the measles and will require hospitalization. About 1 of every 1,000 will actually develop encephalitis, inflammation of the brain, and 1 or 2 out of 1,000 of these people will actually die, even with the best care possible. When you start to think that this is considered a childhood disease, this just has such dramatic outcomes that we really do need to make sure that people are getting their MMR for their measles, mumps, and rubella, which is the German measles. Here's just a little story. This is Alba, who her mom wants to make sure that people understand what measles did to Alba. It almost killed her. She was too young to be vaccinated. They don't know who she was exposed by to get the measles, and that's the problem. When people choose not to vaccinate, there's no vaccine that is 100% perfect. The MMR is very good efficacy, probably about 95%, but that means that some people actually are not immune, and so they are relying on herd immunity that if the virus doesn't have the opportunity to infect, it's just not going to be readily out there in the population. So we need both of the fact that for COVID-19, it'll help that there's people that have been infected and now have healthy antibody protective titers, but we also have to get the vaccines going if we want to prevent the next waves that might come next year, as this could be seasonal. So when we look at the vaccines and what they have done. Again, pre-vaccine era, we see some of the morbidity. So we were able to eradicate things like polio and smallpox, deadly viruses, because of our vaccination policies and development of these vaccines. When we talk about our worldwide health, in Uganda, they were able to actually get rid of Hib, the Haemophilus influenza type B meningitis, by doing what is referred to as a 5-in-1 jab. This had not only the Hib, it also had the diphtheria, hepatitis B, tetanus, and the whooping cough pertussis vaccine. So they were able to vaccinate these children for less than $3. Again, it's alarming when you start to look at the statistics. About one child dies every 20 seconds from what would be these type of pneumonia-like diseases. And many of these are going to be, again, preventable. And many of these are pneumonia diseases where availability of various types of treatment is just not available. So that's why preventative measures are important. When we talk about our bacterial vaccinations, we see things like our TDP for tetanus, diphtheria, and pertussis. Sometimes you will see the T first, sometimes the D first. So you see the DTPs and the booster, the TDAPs. These are going to be for preventing tetanus, diphtheria, and pertussis. These are only good for about 10 years, so we do need to get boosters. The meningococcal, the pneumococcal, and the Hib vaccine, these help to prevent both pneumonias and meningitis. So these are those conjugated vaccines where we had to take that polysaccharide and couple it with the protein in order to make it antigenic enough to be able to make these kind of new generation of vaccinations. As I said, though it's not the only thing that we vaccinate to, obviously you see that there's a number of viral vaccines. We have our annual flu vaccine that is recommended, the classic of our MMR, our measles, mumps, and rubella. Chickenpox is now one that we vaccinate to. Polio. You only get rabies unless you are someone who is at high risk. So you're a vet or you work with wildlife. That is not lifelong immunity. It only lasts about 2 years, so you have to get that every 2 years. We also have the hepatitis B and A vaccines as well, and our newer ones of things like our herpes zoster and our human papillomavirus. Smallpox is available, but because we consider smallpox to be eradicated, it is a little controversial of, do we want to still be vaccinating? Some military and first responder personnel may still be vaccinated because we are concerned about potential bioterrorism, but we have not seen a case of smallpox since 1977. This is the recommendation for your childhood vaccinations. I don't put this here for you to memorize. I will not test you on this, but this is just an FYI of looking at when we start our vaccinations for the various childhood vaccines. Some of the newer vaccines, the herpes zoster is now being recommended for adults over 60 to prevent shingles. So if you have grandparents, you want to make sure that they are aware of these types of vaccines. It is also recommended that those over 65 get the pneumococcal vaccine. That has been a recommendation since 2014. And you are probably aware of the recommendation for the HPV vaccine. Human papillomavirus, not only does it cause genital warts, the concern is that it can cause cancer. Uterine and cervical cancer are the highest risk. And fortunately, there are potentially limited number of strains. HPV 16 causes about 50% of the cancers, so we can make an effective vaccine. The recommendation is for all females ages 9 to 26 to get the vaccine. It's recommended for boys as well. Usually this starts at 11 or 12 years of age to get the first of the two of the series. We're not sure about the duration, at least 6 to 8 years, but the CDC has been following the vaccination for over 10 years now, and there's no evidence of weakened protection over time. So that's why they have their campaigns of trying to educate people that the HPV vaccine is cancer prevention. Ultimately, we would like to have a vaccine that is affordable, heat stable. You have to think about vaccinating the world, getting to villages in places like sub-Saharan Africa. You can't get refrigeration to those areas. And so this also means that we don't want to have things that have to be injected, so orally administered. In the first place, nobody likes to get a shot, so some of these edible plant vaccines have been of interest. Ideally, a multiple antigen. Right now, we pretty much have a 5-in-1 jab, so putting everything in one, it's not realistic at this point, but again, we're talking about what would be our ultimate goal, and a single immunization at birth. Are we there? No way. We're not even close, but we do want to make sure that people are thinking about the need for vaccines. People do not recognize what these diseases may look like if they start to come back, and so it's important that we educate. Part of the problem, the controversy of vaccines and autism. Do vaccines cause autism? No, absolutely not. That has been scientifically demonstrated. The problem is the fraudulent work that was put forth by Andrew Wakefield. Once he put that out there, it's amazing that that very small study actually started the whole controversy. It never should have gotten to that level. It was a very small number, and the data was actually fabricated. Unfortunately, once it occurred, trying to get people to understand that's not the case. Part of the problem was that the original MMR had something as a preservative that was referred to as thimerosal. It is a mercury component, but it's in such small amounts. But even after thimerosal was removed from the MMR, it did not change the numbers for autism. Autism and autism spectrum diseases are increasing regardless of the vaccination policies. There are other reasons that we need to look at and do something about the autism epidemic. However, vaccines are not the causative agent. That's why it's important for you to be advocates. And as I said, although I hope there's many things that you remember from 226, knowing and being an advocate about the importance of vaccines is a definite take-home message.